Hi anatomy students, this is going to be an introduction to the histology slide collection that we have here at Unity. Remember, histology is a type of microanatomy and it's the study of tissues. So we're looking at the closely associated groups of cells that come together and make up the tissues of the human body. A lot of times we're not just looking at the living cells, but we're also looking at what they secrete. So the combination of living cells and their secretions will make the form and function of a tissue identifiable. Remember that we can identify tissues in 4 categories. Epithelial tissues are covering and lining tissues. They form our glands as well. And they're identifiable by what we call cellularity. There's no intervening vascular tissue. So you're going to see dense clusters of similar cells. And there's a lot of living cells in epithelial tissue. Connective tissue can be recognized by its sort of vast array of patterns, similar to this tie-dye shirt that I'm wearing for the Unity Girls Cross Country Conference Championship. You can see the tie-dye has lots of patterns, different shades of light and dark, and it's really groovy. Well, that's going to be the same with connective tissue because we're seeing groups of cells that have a lot of ground substance around them and have made a lot of secretions. And those secretions often are protein secretions that help connect and bind us together like glue. So connective tissue can be broken into connective tissue proper, which is things like tendon and ligament, and aerolar tissue that are made up of collagen proteins and reticulin proteins and elastic proteins and the cells that make those proteins. And then they can also be looked at as another classification is cartilage. Cartilage can be broken into 3 categories. You've got hyaline cartilage, which forms like our trachea, and elastic cartilage, which forms scaffolding, like for our ears, and fibrocartilage, which forms like the intervertebral discs of our neck and our back. Then there's also, within the connective tissue, there's blood. Blood is connecting us. It connects us both nutritionally, brings oxygen as well to the cells, and there are living cells of many different types in blood. Lastly, we have bone. And bone is basically what bone is mineralization, right? We've got living cells that take minerals and they produce this hard mineral substance, bone. And bone can be divided into compact bone and spongy bone, and also can be identified as embryonic bone and also endochondral bone. So either bone that originates in embryonic membranes or bone can be originating from cartilage that is ossifying or becoming minerally hard. So that's the 4 types of connective tissue: bone, cartilage, connective tissue proper, and blood. Last, we have muscle tissue and nervous tissue. Muscle tissue can be divided into 3 categories as well. There's skeletal or striated muscle, that's our voluntary conscious control muscle. Then there is your cardiac muscle, which is your heart muscle. And then last, there's your involuntary smooth muscle, which lines your gut and your arteries. Nervous tissue is really what we're going to be looking at is the neurons. Neurons are complex, large cells with an axon and dendrite. They have ability to send and receive signals and transform the electrochemical potential and activate movement and activate response. So nerve cells work in close association with glandular epithelial cells very often because hormones and nerves often work to communicate through cellular communities called the body, right? So without glandular epithelial secreting hormones and without the nervous system transmitting signals, we wouldn't do a whole lot. Well, let's look now at the Unity slide collection, and I'll take you through and show you different slides that we have here and what they mean. Before we go under the microscope, I want to share with you my favorite slide. I got my classroom in the year 2001. I've been a teacher now for 20 years, and this slide has been my faithful companion through the whole journey. This is a slide of aerolar tissue that was stained beautifully, probably right around the 1980s, I would guess, although it's really hard to tell. I was probably a teenager when this thing was stained. And this has remarkable histological staining properties. And so let's look at aerolar tissue first. Aerolar tissue is connective tissue proper. When I observe these, I'm always going to start on a low power, and I'm going to use my coarse focus to find my target. Once my coarse focus is tack sharp, I'm going to switch over to medium power. I'm on medium power. At that time I can use my aperture ring also, my light aperture ring, to change my settings for observation. In this case I like to dim down the settings just a little bit because I can really see those fine fibers. If I like what I see now, I'm going to switch over to high power. I never go to high power until I've been successful with low and medium power. And wow, this looks really nice. When looking at connective tissue proper especially, but really all cells in life, you have to think of a cell as an artist, and the living artist is going to make non-living art. Think of Claude Monet. Claude Monet passed away a long time ago, but you can still go to a museum and see a Monet. Monet is not a living artist. A Monet is a painting that Claude Monet made long ago. Similarly, when a mammoth rots out of the side of a glacier in Siberia, you can still see the mammoth, but the cells are gone. What's left of the mammoth is all the protein fibers, many of which, of course, were secreted by the living cells of the mammoth as it grew and lived. Mammoth hair, mammoth toenails, the remains of the bones, all the mineralization there, the protein fibers of the tendons and ligaments, they're all still in place. Those are all secretions by living cells, but those aren't alive. Those are non-living secretions. You're looking at art that was made by a living artist. The cell is the living artist. The remains are the art. The cell is the living artist. The remains are the art. The cell is the living artist. The remains are the art. My fingernails, my hair, what's left of it, my skin are all heavily made of keratin. Not living cells, but secretions by living cells. My skin cells are like eccentric artists who have walled themselves up in their artwork to the point where they could no longer nourish themselves and died. A skin cell, a flattened squamous cell in our skin, basically secretes keratin and entombs itself and dies. And so the upper layers of your skin are keratinized cells. So when you look at me, between hair, nails, and skin, what you're really seeing is keratin. You're seeing the non-living covering of my living cells. This is bone, and this is decalcified bone. And it's from a small animal. This isn't from a person. But what you see in the middle is the marrow. Let's get out to the mineralized side of things. That's the mineralization right there. And you can see these little holes. These are called lacunae. That's the places where the cells were living. There's osteoblasts, which make bone by depositing minerals, and then osteoclasts, which are the bone-eating cells. You can also see the rings on there. But this main center of activity here, and we'll look in the middle here, this main center of activity is where the bone-making cells were busy laying down the mineral. That's bone. Let's look at some bone that still has the calcium. There you can see those little lacunae. I like to think of each one of those as a little studio where the artist once sat. And what did the artist do? In this case, the art that was being secreted, the non-living art, was the calcium mineral deposits that we call bone. So these little pink gems here are the smallest cells in the human body. They're erythrocytes, or the red blood cells. You can see a larger cell there that's stained in purple, and that's a white blood cell, one of the immune defense cells. And this immune defense cell has its work cut out for it, because you can see those tiny little purple dots are plasmodium, the protozoan parasite that results in malaria. So this is fairly normal human blood, but it's suffering a plasmodium infection. So these are the main components. You'd expect about one white blood cell for every 600 red blood cells. That's about the normal ratio. Well, unfortunately, this slide here would not be good news. Look how the white blood cells have proliferated and how greatly they exceed the expected percentage. This is, unfortunately, this is chronic granulocytic leukemia. So it's a form of blood cancer where the cells, the immune cells, your hired killer cells, tend to not get the apoptosis signal. They don't get the signal to die. And so they begin to proliferate. There's too many of them. And that starts to gum things up in the capillaries, and the person begins to be starved of oxygen and become deprived of the normal blood functions. And that can lead to death. So this is chronic granulocytic leukemia. This is pretty cool right here. The pointer is on a ciliated columnar epithelial. So we would actually say this is pseudostratified ciliated columnar epithelial, whereas the pseudostratified means falsely stacked. And they all have the same basement membrane basal attachment, but the cells are of different heights, so they kind of overlap each other. And here you can see they have cilia. In the lumen, that open space, they have cilia. And in this case, it's bronchial tissue. This is the cilia that would move mucus out of the bronchial tubes and get it to our cough reflex. So right there is the pseudostratified ciliated columnar epithelial. Now, since this comes from a trachea, we also expect to find hyaline cartilage. So I'm going to explore over here, and look at that. This is cartilage right here. This is hyaline cartilage. And the hyaline cartilage is recognizable. Hyaline refers to its glassy appearance. And the cartilage, of course, is that springy stuff. I wouldn't encourage you to do this too forcefully, but if you grab hold of your own throat, you can squeeze your trachea and feel it springy. And the reason it's going to be springy is because you have hyaline cartilage there. So what cartilage is are these living cells living within their little art headquarters, their little lacunae, their little vessels, and what they're doing is they're secreting these fibers. And in this case, this is cartilage. It's a springy secretion of fibers from these chondrocytes. Chondra referring to cartilage, chondrocytes referring to the cells that make the cartilage. So this is a great slide because we have both a connective tissue in the form of cartilage, and then over here we have that pseudo-stratified ciliated columnar epithelial covering and lining epithelial. I think the most dramatic cartilage to look at is elastic cartilage, and that's the cartilage that forms the scaffolding for your ears, for example. And you can see the fibers. This is a beautiful stain, nice and dark too. I'm going to go into high power now. You can really see there's where the living cells are, but look at the fibers in between there. Isn't that dramatic? So cool. And this kind of helps explain why ears have that rubbery texture that they do. That's the fibers in their elastic cartilage. So I can say fibers and then say elastic cartilage. I'm so tempted to say fibrocartilage, but fibrocartilage is completely different. I'll show you that next. Fibrocartilage forms the intervertebral discs in your body, for example, and also the meniscus in your knees. Fibrocartilage cushions against shock, and you can see that it has these sort of lines of cells that secrete the cartilage. I'm just going to move out a little bit here so you can see the context a little better. So here's fibrocartilage in its context. This is adipose tissue, and you can see those really thin fibers that make kind of a nice springy structure. It's not very strong though, and those support fat cells as well, so that when those fat cells have excess nutrients, they can store them. You can convert extra glycogen, carbohydrate for example, into fat, and those cells will really swell up. So this is glandular epithelium, and this comes from a submaxillary gland from a rat. And so if you look at there, remember the term cellularity? You're going to see a lot of cells jammed together. Let's zoom in here so we can really get a sense for that. Lots and lots of cells that are repeating. Look at all those nuclei. And this is going to be an exocrine gland. This is a gland that's secreting its products out of ducts. And my evidence for that is right here. Look at those donuts there. Those donuts there are cuboidal epithelial cells. And remember, cuboidal epithelial are involved in erosion control, lining ducts. So those products are going to enter into the ducts and be secreted. This is a Masson stain of colon, and these are the goblet cells. So here we're looking at cells that are going to break off at the apical end, and they're going to release mucus And that's going to basically protect and line and provide a slippery surface for the inside of the gut. While it may be easy to think of the intestine as a tube, remember that this is a slice through a three-dimensional world, and the tube isn't smooth. The tube is full of ripples and canyons. So what we're seeing right here is simple columnar epithelial of the gut, in this case the colon. I'm going to zoom in now so you can see those columnar cells. And what you expect to see is that they're all attached to the same basal membrane. The nuclei are all relatively even, and then they've got the other end exposed to the lumen. Recall that the job here of the columnar cells, in this case, would be absorption of nutrients or filtration, or both. Wow, here's a really nice example of pseudostratified ciliated columnar epithelial. And what I like about this slide is you can kind of see the cilia, but you can also see that these cells are truly pseudostratified. Look at how the nuclei of each cell are above and below each other. There you can also see the basal surface. They're all glued down to that connective tissue down there. And what's interesting is you can see some cuboidal epithelial down below there, some tubes. So expect some secretion of products going on. Little glandular epithelial there. And then down here you have hyaline cartilage. So hyaline cartilage is named for its glassy appearance. And this is a great example of hyaline cartilage. Migrating back up, we're into some glandular epithelial here with some ducts, some cuboidal cells. The cuboidal cells are making those donuts. Those donuts are cross-sections through tubes. And then you've got some connective tissue fibers here forming the basement membrane. And there's our pseudostratified ciliated columnar epithelial cells at the top. They're glued down at their basal surface. They're waving their cilia into the lumen at their apical surface, and they're generating current along which mucus can flow through the bronchial tubes. So what a great connection here. Each one of these little fried eggs here with its yolk is actually a simple squamous epithelial cell with its nucleus. So as you're looking at this, you're seeing about 7 or 8 of these simple squamous epithelium that have been scraped off from the inside of somebody's cheek. Simple squamous epithelial refers to the fact that these occur in a single layer, that the fact that they're flat, and that they are covering and lining. Simple squamous epithelial. At long last, welcome to the inside of the kidney. This is where we really get to see simple cuboidal epithelial making that erosion-resistant lining to ducts. Now remember, we're looking at a cross-section of tubes here. We've cut through the tubes and we're looking down the tubes. I'm going to zoom in here and we're going to get to see those cuboidal cells around the lumen. So each one of those donuts, I like to call them donuts, is actually a cross-section through a kidney duct through which urine will flow. And we can see each of these cells are nucleated. They're about as wide as they are tall. This is simple cuboidal epithelial. Simple, occurring in a single layer. Cuboidal, being as wide as they are tall as they are wide. Epithelial, covering and lining. This is striated muscle. And to really appreciate this, we got to zoom way in. And you can actually see those little striated bands. Those bands that run perpendicular to the length of the muscle. Those bands are caused by overlapping motor protein fibers. And so when those overlapping regions, when the proteins gain greater distance from each other, the muscle relaxes. And when those proteins grapple into each other and pull on each other, the muscle contracts. So each one of those bands is part of the solution to how a muscle flexes and relaxes. This is striated muscle or skeletal muscle, and it's found basically as all the voluntary muscles of the body, in our biceps and our forearms and our legs, everything that we use to flex and move. These are the major muscles of locomotion. This is cardiac muscle, and cardiac muscle is a close kin to skeletal muscle. It has striations, You can see that. But it also has a lot of characteristics that are different. For one, the muscle fibers are a little bit thinner, but also there's these interruptions that you can find throughout. Those interruptions are called intercalated discs. And what's really cool is the heart muscle fibers will actually flex on their own. They're coordinated by some nerve clusters, the sinoatrial and the atrioventricular nodes. And so the cardiac muscle movement and coordination in the beating heart is orchestrated by nervous activity. And this is smooth muscle. Smooth muscle, you can see, doesn't have the striations. This is the involuntary muscle that lines our guts, and also the involuntary muscle that lines our arteries. Think about this. Involuntary means it's not under conscious control. It's responding to other feedback, including chemical stimulus and nerve response to other things happening in the body, and goes beyond our conscious control. That's why you're sitting in an elevator and it's all nice and quiet. There's one other person on the elevator and all of a sudden a little bit of gas starts moving through your intestine. It can be pretty embarrassing because you can't control that. You're just going to hear a sound. This is smooth muscle. Smooth muscle is not under conscious control. It lines our stomach, our intestines, our blood vessels. Smooth muscle. Note the lack of striations. I was a little nervous that I wasn't going to be able to find a good slide. Yeah, I did that on purpose. Nervous tissue. This is nervous tissue. And here you can see the specialized nerve cells. These are called neurons. And you can see they're branched. You can see that they have kind of like a tail. And they have like sort of thin little tendrils sticking out, right? Almost like little tentacles. This is a neuron. A neuron has a couple of parts. It has the axon and the dendrite. In dendrite, think about dendrology, the study of trees. A dendrite is a branched structure. So these are neurons from the nervous tissue, and they have an axon and a dendrite. So the very small nucleated cells that sort of surround and insulate these neurons are the neuroglia. So you have the large neurons and then the small neuroglia. What's really cool about neurons, the large cells, is that, generally speaking, large cells don't do well in biology. There's too poor of a relationship between the surface area of the membrane and the volume. But when cells get larger, if they become multiply branched and they increase their surface area through these sort of tendrils, then they can sort of compensate for their size by increasing the surface area to volume ratio and making things run more efficiently. So if you don't remember anything else from this, I hope you remember this. Living cells are like artists, and the products they secrete are like the art. Our bodies are held together by the secreted products of living cells. Those protein fibers of our connective tissue proper are holding us together. When you look at an organ, you're looking at tissues. That tissue is made of both the living cells and the non-living secretions. That's a good way to think about histology. I hope that's helpful.